Hey, welcome! Whether you're trying to nail down some concepts for a biology final or you're just really curious about how life works, you've come to the right place. Today, we're going deep inside your cells, into the genetic control room, to figure out how they make these huge life-or-death decisions every single moment.
All right, let's jump right in with a huge question. Every single cell in your body, from a neuron firing in your brain to a muscle cell pumping in your heart, has the exact same DNA blueprint, the same code. So how in the world do they end up looking and acting so completely different?
Well, the secret isn't in the blueprint itself. It's all about how each individual cell decides to read that blueprint. And that, right there, is where things get really interesting.
And the name of the game here is differential gene expression. It's a fancy term, but the idea is simple. Think of your DNA like a gigantic cookbook with recipes for everything your body could ever make. A heart cell doesn't read the whole book. It just opens up the chapter on how to be a heart. A brain cell, on the other hand, only cares about the brain recipes chapter. Same book, different pages.
So that begs the question, right? Who's deciding which chapter to open? What's actually inside that cellular control room, flipping these genetic switches on and off? Let me introduce you to the transcription factors. These proteins are the master operators of the whole system. They're designed to physically grab onto the DNA at just the right spots, kind of like a key fitting into a very specific lock, and from there, they can either hit the gas on a gene or slam on the brakes. And when you boil it all down, it's this beautifully simple system. You've basically got two types of operators. You have your activators. They're the green light. They say, go, and crank up a gene's activity. And then you have your repressors. They're the red light. They stop things in their tracks, blocking the gene from being used. It's this constant dance between on and off that lets the cell do its job perfectly.
And this is how the outside world can literally talk to your genes. So imagine a hormone shows up. It's a signal from somewhere else in your body. It docks onto its receptor, which is a type of transcription factor. That simple act of binding causes the receptor to change its shape, which physically kicks off the repressor protein and calls in an activator protein to take its place. And just like that, a signal from outside the cell has reached all the way into the nucleus and flipped a genetic switch from off to on. It's incredible.
But let's say a gene does get turned on. The cell's control systems don't just stop there. Oh no, there's another layer of control. Think of it like a quality assurance step that can intercept the message after it's already been sent out.
We're talking about RNA interference, or RNAi for short. And this is a seriously powerful tool for the cell. Instead of blocking the original DNA blueprint, RNAi is more like a heat-seeking missile. It hunts down the temporary instruction copy, that's the messenger RNA, and destroys it before it can ever be used to build a protein. So you're not silencing the source, you're silencing the message.
And the way we figured this out was a complete and total accident. Back in the 90s, scientists were studying these little worms, and as part of a control experiment, they injected some double-stranded RNA, which they thought wouldn't do much. But whoa, were they wrong. It caused this incredibly powerful targeted shutdown of a gene. They'd accidentally stumbled upon this natural, high-powered machine that the cell uses all the time.
So here's how that machine works. First, an enzyme with a great name, dicer, acts like a paper shredder. It finds that suspicious double-stranded RNA and chops it up into these tiny little pieces. Then one of those tiny pieces gets loaded into a protein machine called RISC. Now, that little RNA fragment is the guide. It leads RISC on a search and destroy mission through the entire cell, looking for any messenger RNA with a matching code. And when it finds one, RISC acts like a pair of molecular scissors and just snips it in half. Message silenced.
Okay, so all of this intricate machinery, all these switches and silencers, it's an amazing system. It's elegant, but it's also delicate. And that brings us to a really critical question. What happens when this incredible control system breaks? Well, in one word, cancer. I mean, that's really what cancer is, at its most fundamental level. It's a disease of broken gene regulation. It's what happens when the wrong switches get jammed in the on position, or when the emergency brakes completely fail. The cell loses all control and just starts dividing and dividing and dividing. And this kind of breakdown usually happens in one of two major ways. First, you could have a mutation in what's called a proto-oncogene. That's basically the cell's gas pedal. Just one bad mutation can jam that pedal to the floor. The other way is to mess with the tumor suppressor genes. Those are the cell's brakes. Now, for the brakes, you've got a backup. So you actually need to have mutations that break both copies of the gene. You have to cut both brake lines before the cell completely loses its ability to stop.
So with all this complexity, thousands of genes turning on and off all the time, how can scientists possibly know what's going on? How can they actually get a look inside that genetic control room and see which switches are being flipped?
They can do it using a field called transcriptomics. It's a way to study all of the RNA messages a cell is producing at once. With tools like microarrays, researchers can take a single snapshot that shows them the activity level of thousands of genes all at the same time. It's like getting a direct readout from the cell's entire control panel.
And a result might look something like this chart right here. You can see the story of the cell immediately. In a cancer cell, that proto-oncogene, the gas pedal, is running way too high. And at the same time, the tumor suppressor, the brakes, is running dangerously low. It's a crystal clear signature of a system that's gone haywire.
But here's what's really humbling and kind of mind-blowing. Even with all these amazing tools that let us peer into the cell, we've barely scratched the surface. As Professor Pasquinelli points out, for the vast majority of our genes, we still have basically no idea what they actually do. It's a massive unexplored territory right inside our own bodies. And so, that leaves us with this final, really powerful thought. Understanding this whole system isn't just for a final exam. This is the future. As we get better and better at reading that genetic control panel, the next great frontier in medicine will be learning how to rewrite it, to go in and fix the faulty switches, and maybe, one day, to cure the diseases they cause.